Hello, I'm Bob Goldman, and in our previous talk, I discussed the cytoskeletal intermediate filament networks of cells, and now I'd like to introduce you to the nuclear lamins, which are the type 5 intermediate filament proteins, which many consider to be the building blocks of nuclear architecture. And first, right on this introductory slide, you can see the lamin network, both forming an intense ring around the edge of the nucleus, and in fact forming in lesser concentrations a network throughout the nucleoplasm. As I mentioned previously, nuclear lamins are type 5 intermediate filament proteins. They are encoded by 3 genes: the nuclear lamin A gene, B1, and B2. And lamin C is an alternate splice product of the lamin A gene. So we have both lamin A protein, lamin C, lamin B1, and B2 in the nucleus of most vertebrate cells, certainly human cells. Let me just go through the complexity of the intermediate filament processing, post-translational processing. Before I do, I just want to mention that there are, in addition to the central rod domain, which is slightly longer than the rod domains of proteins like the vimentin cytoskeletal protein, there is an IG fold motif and also a nuclear localization signal in the C-terminus of the protein, in addition to a very C-terminal CAX box. And it is this CAX box that goes through numerous modifications during processing from pre-lamin A to mature lamin A. In particular, what I tell you is going to be true of lamin A, but not 100% true of lamin B1 and B2, and I'll tell you the difference in a minute. And you'll see why later in the lecture I'm going through this complex series of changes that take place. So the CAX box is a signal for an enzyme farnesyl transferase to transfer a farnesyl group lipid moiety to the cysteine residue in the C-terminus. And this is a signal for an endopeptidase called the AX endopeptidase, which removes the 3 amino acids at the AAX at the C-terminus. And this is a signal for the carboxymethylase to then place a methyl group on the cysteine residue. So now you have a farnesylated, methylated cysteine at the C-terminus. And then this is a signal for another enzyme, another endopeptidase, ZIMSTA24, which then cleaves 15 additional amino acids upstream from the C-terminus. So all of this processing goes on and then removes the farnesylation site at the C-terminus. But in the case of lamin B1 and B2, these are both permanently farnesylated, and this is not in their mature form. Now the lamins assemble in the nucleus into a complex series of structures, but let me just tell you where it resides in the nucleus as seen here in the electron microscope. Just under the nuclear envelope, and if we look in the next image at high magnification, the lamins you'll see are forming an interface in a region called the lamina between peripheral heterochromatin and the inner membrane of the nuclear envelope. And here you can see it in this famous picture by Don Fawcett. You can see the outer nuclear envelope membrane, the inner membrane, the outer membrane, the inner membrane, and this fuzzy fibrous layer, which is called the nuclear lamina, here beautifully seen, rarely seen in this thickened configuration. But then you see just inside this lamina region, the peripheral elements of chromatin. So the nuclear lamina it forms a molecular interface between the inner nuclear envelope membrane and peripheral chromatin, peripheral elements of chromatin. The lamins, it turns out, are the major proteins of this lamina region. Lamins, as is the case with vimentin, form dimers, and these dimers interact in a way that's quite different from vimentin. That is, they interact by what's called head-to-tail assembly of dimers. So dimer interactions take place rather than the kind of lateral association we see and longitudinal associations we see with vimentin. Here we form dimer-dimer interactions into long chains called protofilaments, which then aggregate laterally to form higher order structures, ultimately resulting in the formation of these beautiful structures originally described in Xenopus oocyte nuclei by Uli Aby, where you see almost a lattice work, almost a square lattice work of lamin filaments. The nuclear lamins have structural properties and functions which are not dissimilar from the intermediate filaments in the cytoplasm, but now we know many more specific possible functions for these proteins in the nucleus. And they certainly are major structural proteins of the lamina, which is surrounding the edge of the nucleus. They're also located throughout the nucleoplasm. And we know that the determinants of nuclear size and shape, we know that nuclear envelope assembly and disassembly is regulated to a great extent by the nuclear lamins, and this has been shown by numerous laboratories. They are involved in the formation of the mitotic spindle. They're involved in DNA synthesis, specifically at the chain elongation phase of synthesis. They're involved in DNA damage repair. They have a role in transcription, especially RNA pol II transcription. They're important in cell proliferation and senescence. They provide structural support for the nuclear membranes and also support in positioning of nuclear pores. And they provide chromatin anchorage and organization sites in the interphase nucleus. I want to tell you a few of these, give you a few pieces of experimental evidence that what I told you, at least some of the things I told you are correct. And that is, if you design a dominant negative mutant of lamins, as we did quite a few years ago, by removing the head domain, you can see in this second diagram here that we've removed the N-terminus of lamin, and then this interferes in some way with lamin assembly, because if you take this protein, express it in bacteria, and this was human nuclear lamin A, so we call this headless lamin A or delta NL-A, and inject it into a cell which is expressing GFP, so there's a live cell expressing GFP lamin A. On the far left, you can see the cell before injection. In the same cell, 30 minutes later, you can see that the lamins are beginning to form foci. They actually disassemble at the nuclear surface and form foci throughout the nucleoplasm and become significantly misshapen within 1 hour to 90 minutes, 1 to 1.5 hours, as you can see here on the right. And we noted that under these conditions where you get disruption of lamin organization, you get not only lamin A disrupted in this case, but also lamin B. And lamin disruption also alters nuclear shape and the mechanical integrity of the nucleus, and also stops DNA replication, as I'll show you in a minute, and RNA pol II transcription. In the case of DNA replication, although we can show this in cultured cells following microinjection of single cells, it's much easier to use the Xenopus laevis cell-free nuclear assembly extract, which you make from Xenopus eggs. And basically what you do is in order to do biochemical assays, this allows you to make hundreds of thousands of nuclei very rapidly in the test tube. And on the left side, you can see sperm chromatin, which has been denuded. That is, the sperm tail is removed and the membrane in the head of the sperm is removed, so all you have is a condensed mass of chromatin, and you put it into this interphase extract, which is easily prepared by centrifuging Xenopus eggs. And then you get nuclear envelope assembly and decondensation of chromatin around each of the sperm heads. And this just shows nuclear lamin staining around the edge. In the case of Xenopus eggs, this lamin is called lamin B3. And using these nuclei, as soon as they become enveloped by the lamins and the lamina in a nuclear envelope, they initiate DNA replication. And if we take these nuclei at that time and just expose them to the delta N LA or delta NLB or delta NLB3 from Xenopus, any of these will work. It will disrupt nuclear lamin organization in these in vitro assembled nuclei seen over here on the left in the middle panel. You can see that nuclei become misshapen and the lamins tend to form aggregates as we showed in the microinjected cultured cell. So here we can make lots of nuclei and you can see that the lamins are disrupted into these aggregates which appear throughout the nucleoplasm. And at the same time, if you look at the incorporation of P32 labeled ATP, which is a precursor or to looking at precursor to looking at DNA replication, you can show that in a dose-dependent way, DNA replication is inhibited, and you can see that throughout this series of agarose gels. So DNA replication can be inhibited in a dose-dependent way by adding this dominant negative mutant of lamin assembly, and in fact, you can basically reduce the replication by as much as 95% with this lamin mutant. But you always have residual DNA replication, and it turns out that that's because the initiation of replication can take place, but the chain elongation phase of DNA replication is inhibited. And this is totally recoverable because if you put these nuclei back into normal medium, they'll begin to replicate their DNA. And one of the features we looked for in these disrupted lamin nuclei were changes in two replication factors. One is replication factor C, seen in this top series, and the other is replication is PCNA, proliferating cell nuclear antigen, which is both very important factors in the chain elongation phase of replication and in fact, and the function of DNA polymerase delta. And you can see here that both of these factors seem to line up or co-localize with the aggregates of lamins that form upon disruption of the lamin network. And this, of course, is double indirect immunofluorescence as an assay. On the top is the upper left and lower left are the controls for these experiments. If you then look at the regulation of transcription, in this case RNA pol II transcription in HeLa cells, what we did was to microinject on the left here in panel E. You can see what happens after injecting this truncated form of lamin in a nucleus that was fixed about a half hour after injection. This one on the right was injected at a slightly earlier time. And you can see in both cases there's disruption of the lamin organization, but the one on the left is even more disrupted. And then on the right, you can see the result of the same nucleus which was injected and incorporated or exposed to BRUTP, which is a probe for RNA synthesis. So you can see that there's a graded response. This is greatly reduced in BRUTP incorporation, and this is less reduced in BRUTP incorporation. And on the bottom, you have a third cell nucleus, which was not injected at all with this dominant negative mutant. So there is a great reduction in RNA synthesis as indicated by BRU incorporation. And on the bottom, it just shows that there's always residual BRU incorporation. It's always associated with nuclei, which you can see in the center panel in phase contrast, which indicates that RNA, actually the data show that RNA pol I and pol III are both going on normally. It's only pol II that's inhibited. And then we repeated this same experiment with transcriptionally active isolated nuclei from Xenopus embryos. The Xenopus eggs were fertilized and embryos were allowed to develop for 10 hours into the mid-blastular transition, which is the time when RNA polymerase II becomes most activated. And you can see in the panel C that transcription of messenger RNA is greatly reduced compared to D in the same gel. These are agarose gels showing, again, P32 UTP incorporation into RNA. The band at the bottom of these gels, in fact, is transfer RNA, which is synthesized, of course, throughout these early developmental stages. But in the case of looking at nuclei, the nuclei that were isolated, you can again show that when delta NLA or delta NLB3 is incorporated into these nuclei in vitro, you get disruption of lamins, and again, lower right shows that BRU is not incorporated. So it looks like transcriptional control of RNA pol II is in some way regulated by a normal lamin network. Now I'd like to turn our attention in the last part of this talk to what rare human diseases have told us, and in fact provided an enormous number of unexpected insights into nuclear lamin structure and function. So it turns out that in the last decade, or slightly over a decade, a number of, in fact, a rather enormous number of diseases and mutations, now somewhere between 300 and 400 mutations, have been related to diseases caused by changes in the nuclear lamin A gene. So this remarkable number began with the discovery of Emery-Dreifuss muscular dystrophy many years, about 12, 13 years ago, by Giselle Bon in France. And it turned out that this was a point mutation in the nuclear lamin A gene. And this was followed by an enormous number of discoveries of mutations in the nuclear lamin A gene, probably making it the most mutated gene, or one of the most mutated genes in the entire human genome, that causes many different diseases. And included in these diseases, as you can see on the left, is the human disease Hutchison Gilford Progeria syndrome, which is a premature aging disease of children. There are also forms of nuclear diseases of neuromuscular junctions called Charcot-Marie-Tooth disease. There are also various forms of muscular dystrophies, and in fact, dilated cardiomyopathies, lipodystrophies, and numerous other types of diseases. The mutations are located everywhere along the protein chain, and a given disease can have numerous sites in different regions, and this is very hard to explain and in terms of the total picture. But if we concentrate on one particular disease, and I want to do that today, and that is the disease Hutchison Gilford Progeria syndrome, the premature aging disease of children. And the reason for concentrating on this is it certainly is the most extensively studied of all of the so-called nuclear laminopathies. Hutchison Gilford Progeria syndrome, or progeria, if one looks at the clinical phenotype, you find that children are born normal looking, and typically begin to lose their hair at 12 to 18 months. This is the same young girl progressing through the disease. These three images are of the same patient with progeria. And you can see the disease usually begins with losing hair or becoming bald, and then skin becomes mottled. There's loss of subcutaneous fat. The nose becomes pinched, and there's abnormal tooth development and premature loss of teeth. Typically, they're short in stature, and they rarely achieve a height of 100 centimeters. They have terrible problems with arteriosclerosis, osteoporosis, hip dysplasia, and typically they die at a very early age, usually 13 to 14 years, of heart attacks and strokes. And on the lower right here, we see a typical occluded artery from a patient, a progeria patient. Now, most of the mutations, most of the major cause of progeria is a mutation which is a silent mutation, G608G, which introduces a cryptic splice site, or makes available a cryptic splice site. And this results in the loss in pre-lamin A of 50 amino acids between the end of the C-terminus, just before the Cax box, and 50 amino acids upstream. And this produces a protein, which is called progerin, and it's a heterozygous situation, so therefore there's one normal allele, there's wild-type lamin A as well as the mutant lamin A expressed in these patients. So because of this truncation, the protein that's produced, progerin, is in fact permanently farnesylated, because that first cleavage site is retained normally, but the second cleavage site, which is farnesylated, which should remove the farnesylated methylated C-terminus, in fact, is not available. So the Zimsta-24 can't work, and therefore you get a permanently farnesylated form of mutant lamin A working together with its normal wild-type allele. That's the most prevalent form of progeria, accounts for 70 to 80% of all the patients known. There are other very rare mutations in different regions of the protein, but in fact this is the one that's most common. And it turns out that years ago we found that that if one looks at the nuclear lamins in these patients at early passage skin fibroblasts, one finds here at passage 6, 13, and 26 of the same patient cell that in fact the nuclei become progressively misshapen. So one of the hallmarks of progeria that's been widely used is the formation or the dysmorphology of the nucleus. And these nuclei are called bleb nuclei. And you can see in the middle panel here, at panel F, that in fact there's an increase in the truncated form of protein. This third band appears with a lamin A antibody in a Western blot of a whole cell extract. You can see normally at passage, early passage 9 here, that in fact you see lamin A and C with the same polyclonal antibody. But as you get old, as you pass the cells in culture, or they get older in culture, you see the addition of this third band, which is called progerin. And one other point that I want to make is that we noticed very early on in late passage cells, which are highly blebbed, with highly blebbed nuclei, that in fact there's a separation between lamin A and B. Here lamin A is seen in red by immunofluorescence, and the B-type lamins, or B1, B2, are seen in green. And then if one looks in the electron microscope at these bleb nuclei, on the far left, you can see a typically misformed nucleus of a progeria patient's skin fibroblasts. And in the middle panel, you can see that, in fact, the lamina, this very dense black line here, is greatly thickened compared to a normal control, here seen way over on the right. And there's very little peripheral heterochromatin in these nuclei. In fact, seems to be a great reduction in heterochromatin throughout the nucleus. So we turn to female cells, both normal female and progeria cells, to show losses in heterochromatin because of the presence of the inactive X chromosome seen on the upper left panel stained with histone mark, histone H3, lysine 27 trimethylated. And the same mark you can see disappearing as a function of passage number in progeria cells in the lower left two panels. In the middle, it shows exist RNA, another mark for the human inactive X chromosome, and you can see as you go down through these panels that in fact the exist RNA seems to be retained, but it sort of unravels. It's not as tight a structure and appears to move interior into the nucleus. And over on the right, it just shows you these at higher magnification. On the lower right, you can see the result of an RT-PCR analysis, which shows that the enzyme EZH2, which is responsible for the H3 K27 methylation, is greatly reduced in amount and as a function of passage number again. And then on the left, in the lower left, we see that EZH2 is also greatly reduced in the progeria cells at later passages. And on the right, you can just see another histone mark, H3K9, ME3, also greatly reduced by Western blotting in progeria nuclei at later passages. And you can also see in the nuclei of the patients that there's very little peripheral chromatin, heterochromatin containing the H3 K9 ME3 mark compared to controls. So there are profound changes in heterochromatin in progeria, and in fact, this taken together with the finding that that progerin is permanently farnesylated, and in fact seems to be a dominant negative effect in these patients. This led to a very rapid, probably one of the fastest bench-to-bedside drug trials in the history of medicine, because there were available farnesyltransferase inhibitors, and this attracted participants from all over the world. Parents brought their kids, and still bringing their children, progeria patients, to Boston Children's Hospital, where these trials are being conducted. It turns out that when you inhibit farnesylation of progerin by adding FTIs in the nuclei of patient cells, as was done originally by the Capel group in Francis Collins' lab, you can see that there's a reversal on the lower left of the nuclear morphology. That is, the nuclei look more normal after farnesyltransferase inhibitor treatment. And the nuclei in progeria cells also showed a rather remarkable separation of the A and B type lamins. I showed this picture earlier, but here it is a higher magnification. And you can see in red lamin A, and you can see the B type lamins are excluded from these large blebs, but are present in the other non-bleb region of the nuclear surface. So we wanted to know how this change in nuclear lamin structure might be achieved. And to do this, we just carried out some typical cell biological experiments, which involved silencing each of the lamins. So we designed silencing vectors, shRNAs for lamin AC, for lamin B1, and lamin B2. And you can see at the top panel here, using Western blots, that these work quite efficiently, and the controls are on the right in each case, and the silenced cells are on the left. And in all cases, in these experiments, we used HeLa cells. And on the left, you can see lamin AC staining in a lamin B1 silenced nucleus, and you'll notice that there are blebs in the silenced nuclei. The red nuclei in the middle are unsilenced cells. This was done by transient transfection, so we always had some unsilenced cells, and when you have these, you see normal lamin B1. But in the case of the silenced nuclei, invariably we saw blebbing at the surface. And this was not true when we silence lamin B1 and lamin A or lamin B2, only lamin B1. The blebs were induced, and we found that they were mainly lamin A enriched. That is, they were devoid of the other lamin, lamin B2, because we knew LB1 was missing in these cells. So LB1 silencing leads to the formation of lamin A rich blebs, which looks very, very similar to the case or to the progeria situation. Then we did some experiments just to begin to see if we could induce histone modifications, and the answer is that in the blebs we saw a great reduction. That is, in these lamin A rich blebs, we saw a great reduction in histone marks that usually stain peripheral heterochromatin. In this case, H3K9 on the bottom panel, Me2 was greatly reduced, almost not detectable in the blebs relative to the main nuclear body, which you can see towards the bottom of these images. Again, this is indirect immunofluorescence throughout. And then we asked questions about the blebs, which had to do with their function or the chromatin related to these blebs. What were their properties? And if you look at the upper left panel where it says DNA in A, you'll notice that the bleb that's present, it's the same nucleus all across the top, is greatly reduced with respect to DNA concentration. This is a Herxed stain, and you can see a great reduction in the chromatin, indicating that there's euchromatin in the bleb and not heterochromatin. So then we looked at RNA polymerase II, and if you look at the inactive form of POL2, you find it's distributed throughout the nucleus and even the nucleoplasmic, and even in the bleb. But then when you look at POL2-0, the active form of POL2, it's a specific antibody, you find that it seems to be concentrated in the bleb region. And then we just ask the question, well, since POL2-0 is concentrated in the bleb, we said maybe it's really transcriptionally active. So again, we use the BRUTP incorporation to look at RNA synthesis in blebs. And if you look at that third row of images, you'll see that BRU is greatly reduced, barely detectable in the bleb, whereas it is detectable in the rest of the nucleus. And then if we look, and you can see LB1, of course, silenced in all of these. Occasionally, a nucleolus would get trapped in a bleb. You can see in the bottom panel. And there you can see that brighter object in the bleb, in fact, is a nucleolus because the ribosomal RNA continues to work in the presence of lamin A. So there's no inhibition of ribosomal RNA synthesis. And then we looked at a couple of other marks for transcription by POL2, and that's H3K4ME3 and H3K36ME3, and we found that K4ME3 is present in the bleb, but K36ME3 seems to be absent in the bleb. And finally, we came to the conclusion, which we're in a situation that we're still working on, and that is, it looks like in the bleb, RNA POL2 can be initiated, polymerase action can be initiated, but it can't be completed. So it looks like a phenomenon called RNA polymerase II stalling, or POL2 promoter proximal stalling. So maybe it looks like initiation of RNA synthesis can take place, mRNA synthesis, but the elongation phase can't take place in this case. So because of the changes in the differences in the silencing of lamin B1 and B2, we started to take a more serious look at the possibility that A and B type lamins really have different functions. And what we had noticed earlier in our progeria experiments, and here's on the left you can see Western blots of another mutation causing progeria, which is E145K. You can see that lamin B1 is greatly reduced when the cells become prematurely senescent in culture. And that is one of the hallmarks of progeria. Cells don't grow that well in culture. Progeria patient cells will grow a limited number of generations. Both with the more common G608G mutation and this E145K mutation, we saw premature senescence, loss of growth, and a reduction in LB1. But if you look down on the left, from both the progeria patient on the left, that panel, you can see a great reduction in lamin B1. Lamin B2 is reasonably retained, and lamin-a-C are also retained. So we started asking questions about lamin B1 during normal cell growth and senescence. And here you can see the result of just beginning to look at WI38 cells, which are classical cell system for looking at the growth and senescence of normal human diploid fibroblasts. So as I mentioned below, to date we've focused most of our attention on lamin B1 because of our results from progeria. And here you can see that the loss of lamin B1 is closely coupled to the appearance of senescent markers in human diploid fibroblasts. So here we have beginning at passage 30, and then we look at passage 39, and then passage doubling, I should say, 41. And you can see that the nuclei from progeria cells as we move across change in different ways. For instance, at passage 30, the lamins look quite normal. Lamin B1, lamin B2, lamin AC have their normal distribution. But by passage 39 and 41, we begin to see a loss in lamin B1. And by 41, you can see almost complete loss of lamin B1. This is accompanied in the lower panel towards the right. You'll see a misshapen nucleus stained with LB2. So the nuclei get larger and somewhat misshapen. And then as we move across, these cells at passage doubling 41 are in fact senescent. And we see the appearance of known senescence markers, including some SAHF, which is SAFs, which are senescence-associated heterochromatic foci. And we also see a loss of incorporation of BRDU, which indicates that DNA synthesis goes down to 0 in the senescent cells, indicating no more growth. And you can also see the increase up to 100% of the cells containing what's called senescence-associated beta-galactosidase. All of these are markers for senescence, and we'd like to add to that biomarker list lamin B1, and this has been verified by several different laboratories now. So we went on to look at other, first of all, quantitative Western blotting of the loss of lamin B1, which we can see in the upper panel here as a function of time. As cells enter senescence, LB1 is lost, but you can see on the right-hand side here that LB1 is lost, but LA, LB2, LA, and LC are in fact retained. And the mRNA, as measured by RT-PCR analysis for lamin B1 in red, you can see in this histogram, is greatly reduced even by passage doubling 39, and by passage PD41 is almost impossible to detect. So both the RNA and protein levels of lamin B1 are greatly reduced in senescence. LB1 silencing also slows proliferation and induces premature senescence in WI38 cells. And we showed this by taking relatively early passage cells, let's say passage doubling 24 cells. And if you look on the upper left, you'll see that LB1 is greatly reduced in the silent cells. And this is correlated with a loss of growth, as you can see here in this growth curve, compared to control, which is a scrambled sequence here instead of the silencing vector. And you can show also that in cells, the nuclei get larger and in fact look senescent relative to scrambled controls, but most importantly, they show a significant increase in senescence-associated heterochromatic foci, a significant loss of BRDU incorporation, indicating that the cells are rapidly entering replicative senescence, and a great increase in the number of beta-galactosidase positive cells. All of this tells us that lamin B1 in some way, or the ratio of lamin B1 relative to lamin B2 and the A-type lamins A and C is critically important in both regulating cell growth, cell proliferation, and in fact entry into replicative senescence. And we are now studying the mechanisms by which lamin B1 acts in this fashion, and the preliminary evidence indicates that it's very important in regulating genes involved in oxidative stress. In closing, I would like to acknowledge and thank all of the members of my own laboratory who have worked with us in the recent past and are presently in the lab, as well as our collaborators from here and overseas. And I'd also like to thank our funding agencies, the National Institute for General Medical Sciences, the National Cancer Institute, the Ellison Medical Foundation, the Progeria Research Foundation, and the Bruce Lipper Foundation. Thank you.